Good morning. Good morning, everybody. Happy Monday. Good to see you. Yeah, please go ahead and sign in. Good morning. Hope everybody had a nice weekend. So, we are in the last week of classes this week. Next week is the final exam. So, it's been a long journey through microbiology, but you guys have done such a great job. I was very pleased with the fourth lecture exam. You did a wonderful job. And here we are in our last week of topics. So for lecture this week, you have two short topics and one regular length lecture topic. You have a lecture that's called Introduction to Vaccination, and you have one that's an introduction to epidemiology, two important topics in public health and healthcare in general, very timely now with the pandemic. And then the second lecture topic, the last lecture topic, is called pathogens and how they spread. So this is a focus on infectious disease, essentially, and some of the features of infectious disease. Again, not comprehensive, but more of an introduction to that important topic. Again, very timely these days. So those are your lecture topics for the week. There's two lecture quizzes. The first one covers vaccination and epidemiology. And the second one covers the pathogens. So that's in lecture. And then, of course, next week, on Monday, you'll have your final lecture exam. Now we'll talk just a little bit about the final lecture exam, because it's not a traditional final exam. Instead of being a review of the course, this final exam is essentially your next lecture exam. It covers the material in lecture since lecture exam 4. So the immune system, vaccination, epidemiology, and the pathogens material. Now you'll notice that the final exam is worth a little bit more points-wise than the other exams. It's worth 100 points instead of 80 points. And that's because there will be a section of the final exam that focuses on old material. In other words, there'll be a section of the final exam that is cumulative. Now, it's not purely cumulative. I'm not going to re-examine you on all of the topics. But what I am going to do is go back through the first 4 lecture exams, and I'm going to pull out questions that more than one person missed. I'm going to go back through and I'm going to find the questions that were difficult for at least a few of you. And those will reappear, not necessarily word for word, but the concept will reappear. in a question on the final. So because this final exam is worth more than the other exams, you can expect it to be a little bit longer. Instead of having about 25 questions on it, there might be more like 30 or 35 questions on it. You still shouldn't have any trouble completing it because you're still allowed to use your lecture notes. But what you will need to do between now and the final exam, in addition to watching this week's lecture material and taking notes, you'll need to go back through the first 4 exams and look at any of the questions you missed and what my comments to you were about what you missed. Because there's a chance that that concept is going to appear again. For example, let me give you an example. On the last lecture exam 4, there was a question about whether or not both strands of DNA are used during transcription. I can't tell you the exact wording of the question because I can't recall it, but essentially the question wanted you to recognize that during transcription, one gene is involved on one strand of DNA, the strand that we call the template strand. And during that process, that gene, that length of DNA, is used to make an RNA, specifically a messenger RNA. So only one strand is used during transcription and only one little portion of one strand. The gene is what gets transcribed. So you may see that question show up on the final in some way, shape, or form. It won't be the exact same question, but it will be the same concept. So about 25 questions devoted to the new material: immune system, vaccination, epidemiology, and pathogens. And about 5 to 10 questions from old exams that more than one person got wrong. You may go back through some exams and you didn't get anything wrong. And if that's where you are, just review the exam, read through the exam. Make sure you feel comfortable. You'll have your lecture notes next to you. Just make sure that you have them all next to you for the whole course. Okay. And again, I don't anticipate anyone will have time trouble because most of you complete exams in about 30 to 60 minutes. And you'll have a full 2 hours for the final. So that's a week from today. It's hard to believe. Now, the other thing I need to remind you of is to go in and look at your grades. Go in and look at your grades and know where you are. You have 2 lecture quizzes to complete this week, and then that final lecture exam next week. And then you have a laboratory task too, actually, this week. You have your last lab practical on Wednesday. And that lab practical will cover the lab topics that we've gone through since the second lab practical. It starts with carbohydrate fermentation, protein catabolism, respiration. So you'll want to make sure all your lab notes are in line for that. It'll look just like the other lab practicals. It's no longer or shorter. It has no cumulative questions on it. It's just the next lab practical. But as you know, these last few biochemical tests that we've been talking about have been the most difficult of the semester. So you'll want to make sure that your lab notes are in really good shape. And just like last time, when you click on the link for that lab practical, there'll be a document that has the questions on it. And you'll need to open that up in a separate window and then answer in the Canvas quiz, just like last time. Approximately the same length. And the second thing that you'll be doing this week for laboratory is your final lab project or your final lab assignment, because it's not really a project. So this week, in lieu of doing lab questions, you'll be doing a lab assignment, and it's called Identifying an Unknown Microbe. So all semester long, we've been talking about what we do in a microbiology lab in order to identify a microbe. We do basic things like gram staining and wet mounts. We make smears. We examine microbes under the microscope. We also culture them in sometimes basal medium, sometimes in chemically complex medium. We just culture them, we grow them. Sometimes we grow them on plates, making a quadrant streak so we can see the colony morphology, because that's a piece of information that tells us something about the microbe. And then there are a whole series of special culture media that can help us select and differentiate different species of bacteria, so we can use those. And then we have a whole series of biochemical tests that we can run, looking for specific metabolic abilities in bacteria. We gather up all this data, and then we go into published source materials and we say, which bacterium has all these traits? And it helps us figure out what bacterium we're dealing with. So that's what your final assignment is. Your final assignment is to take a set of results from all of these types of laboratory procedures and figure out what microbe was tested. What is the genus and the species of the microbe? Now, I'm going to walk us through the assignment, the actual assignment sheet, and I'm going to show you how to use some of the tools that you'll be allowed to use for the assignment. That's what we'll spend our time doing this morning. But this is basically the culmination of everything that we've done in lab. This semester, you're going to get a chance to use this new knowledge you have regarding identifying bacteria specifically in the laboratory, something that happens every single day in microbiology, diagnostic laboratories, and research laboratories. So before we start talking about the assignment specifically, let me ask if anyone has any questions about anything else that's going on. Any questions about the immune system lecture, for example, or if you have started into vaccination, if you have any questions about that. Any questions at all? If they come to you during our time together today, go ahead and type them into chat and we can talk about it. This will be the very last time that we get together on Zoom this summer. So feel free, anything at all you want to talk about, we can talk about. I won't see you on Wednesday because you'll be taking your lab practical on Wednesday. And I won't see you on Monday because Monday is the final exam. And then we're done. We're finished. A good summer's worth of work. All right, so the first thing we're going to do is we're going to look at the module so everybody knows what where to find all of the material I'm going to show you today. So Elaine's asking if there are any slides for today, and the answer is no, Elaine. No, we're all done with our lab procedures. So all we're going to talk about this morning is this assignment. Okay, and it will be due on Sunday at midnight, just like your typical lab questions are due. All right, so the first thing, could you do me a favor? Excuse me just a minute. Could you turn that little fan on for me? Pointed at me, the one over in the living room. The light is very hot. Excuse me, my son is heading out to work, so. All right. 1, 2, 3. All right. So if you go into the module section, and that's what you should be looking at right now, our Canvas page with all the modules in it. Here's the to-do list for this week. The vaccine lecture, the quiz. Oh, I apologize. I've actually posted 3 separate quizzes. I thought I only posted 2, but that's all right because it's extra points for you, extra opportunities to earn points. So intro to vaccination and epidemiology, I've actually separated, but they're both due Wednesday at midnight. And then the pathogens quiz is due Friday at midnight. I apologize. I thought I had combined these, but I didn't. So if you look under Monday's lab, this is where you'll find all the materials that you'll need for this final lab assignment. And then for Wednesday, you'll be taking your lab practical exam. And then here's where you're going to click to submit your final unknown microbe assignment. And again, that's due Sunday at midnight. All right. So if I click where it says materials, it's going to take me to a page. Okay. So let's take a look at this. You are absolutely free to work with other classmates for this if you would like to. But each student has to submit their own assignment. In other words, each student has to write up their own assignment in their own words. Every once in a while, I'll get 2 students who submit identical assignments on this. Don't do that, or neither one of you will get points. Okay, so make sure you write it yourself. Even if you get together with a classmate and talk about it, there's nothing wrong with that. And this final assignment is worth as much as a lab practical exam. So give it the time it needs. Now you've got 3 pieces of information here. You've got 3 resources. You've got the instruction sheet. That's what we're going to look at together. And then there are 2 different data sets. So if your last name begins with A through M, this is the data that you're going to use. These are all the test results that you're going to use and identify the microbe that produced them. If your last name begins with N through Z, this is the data set that you're going to use. So half the class approximately is going to use this data set. The other half the class is going to use this data set. Okay. And then you'll notice there's a document called dichotomous keys. We'll take a look at this document as well, because you're going to use this as source material, and then you're also going to be going onto the internet to some reputable microbiology resources. so that you can take these test results and use them to figure out which bacterium you have, what the genus and the species is. All right. So that page with that material is in the modules section. That's where you'll find all of it. But when you're ready to submit your work, you're going to go to that link that says submit your assignment here. You have to send it in through that link. Okay. Oh, so Valerie has a question about the immune system. Very good. Is antibacterial gel bad for your skin? I know your skin is important to your immune system. Is it better to just wash your hands? What a good question. And this is a question I get a lot from people, especially during this pandemic. I want us to remember what the skin does for us. Skin is a couple of important points for us in terms of our immune system. First of all, it's a physical barrier. It's not possible for microbes to just crawl through your skin. They cannot do it, okay? So it's a physical barrier. It's a wall to things crawling into your body, okay? But you have lots of holes, don't you? You have holes and you have holes and you have all kinds of holes where bacteria can get in. All right. So your skin is also covered in a microbiome. Your microbiome is a living barrier to infection. Your microbiome, all those microbes that just live on you, actually produce a barrier to infection, because if a pathogen gets on your skin, and it happens all the time, if a pathogen gets on you, it simply cannot get through all those other bacteria because they have established a community. Remember, we talked about biofilm and the fact that the microbiome is tightly attached to you. You can't wash it off. So you have a living barrier on top of a physical structural barrier in your skin. It makes such great protection for us. So the question sometimes is, well, gosh, maybe I shouldn't put an antibacterial material on me because it will kill my helpful microbiome and it will get rid of one of my helpful barriers. Here's the answer. Oh, and of course, the second part of the question is maybe I should just use soap and water. I should avoid antibacterial things and I should just use soap and water. So here's the most important thing to know. Soap is antibacterial. Soap is antibacterial. We don't talk about it as an antiseptic or a disinfectant on surfaces, but soap is able to disrupt membranes. That's how it works. It breaks apart membranes. So the reason soap and water works really well to keep you from getting sick is because soap itself is antibacterial. Now, most people don't wash long enough. Remember, we talked about how it's very easy for people to misuse disinfectants. It's also really very, very common not to use soap correctly. You really do have to soap and rub for about a minute to 2 minutes. You really have to do it for a while. But it works. And that's why, for many of us, part of the reason we're not sick all the time is because not only do we have a functioning immune system, but we also wash quite a bit with soap. So here's the other important thing to know. Most pathogens are planktonic bacteria. They are free living bacteria. Remember, they're the kind of bacteria that are in our microbiome and are literally adhered to us. And then there are the kind of bacteria that are planktonic. They are free living, and we pick them up all day because we touch things and we bump into things and we hug people. We pick up little individual bacterial cells all the time, and some of them are pathogens. Antibacterial soap and plain soap that does not claim to be antibacterial, those are good at getting rid of planktonic bacteria. They're not going to harm your microbiome if they're used correctly. So if you're using an antibacterial soap to wash your hands, you're not going to harm your microbiome. Your microbiome is really tough because it's in that sticky film. However, it is possible to misuse antibacterial preparations. There are people that not only bathe with an antibacterial soap, but that scrub themselves like with the kind of sponge that you would use on your dishes. There are people that scrub themselves raw when they bathe. Those people may be harming their microbiome. The funny thing is, you can't get rid of it entirely. Even the folks that really have a psychological problem and wash all day, essentially, they're never going to get rid of their entire microbiome. But the reason it can be harmful is because if you wash too much with an antibacterial product particularly, there's a danger that you're going to not disrupt the microbiome so much, but you're going to disrupt the skin. So the reason it can be problematic is because it's very drying to overdo it with soap. If you're the kind of person who showers and scrubs themselves until they're red, you are literally creating micro fractures in your skin. If you're the kind of person who washes their hands excessively, and I'm talking, on the order of, say, an obsessive compulsive level of washing, your hands are going to be raw. And it's not, again, so much that you're harming your biome, but you're harming your skin. Antibacterial soap can do that, and regular soap can do that. So whether you use one or the other, it's really just a preference. It's just a preference which soap you prefer. Both of them are perfectly safe if they're used correctly. Wash your hands multiple times a day for 1 or 2 minutes. Bathe once a day, approximately, but don't try to scrub your skin off. Because if you damage your skin with excessive bathing, that will allow planktonic bacteria to get in. Remember, the skin's a beautiful barrier, but if it's disrupted, if you've got literal micro cracks and bleeding, that's a way for microbes to get in. It's such a good question. During the pandemic, a lot of folks were sent to hand sanitizer, right? The idea was we didn't know how this virus spread. We didn't know its aerosol droplet nature right at the beginning. So we were concerned that people could pick it up off a doorknob or off their groceries. Remember how scared everyone was about groceries? And put it in your mouth and get COVID. So everybody went crazy at first with hand sanitizer. Some people were spraying down their Amazon packages because we were so worried at the beginning, and rightfully so. Very scary to go through a pandemic. So a lot of people have adopted the habit of using hand sanitizer. Now, what's the primary ingredient in hand sanitizer? Type it into chat for me. What's the primary ingredient in hand sanitizer? Carrie's got it. Marilyn's got it. Yun's got it. John's got it. Alcohol. It's alcohol, right? And as we know, alcohol is rapidly bactericidal when it is used as either a disinfectant or an antiseptic on surfaces or on skin. It's rapidly bactericidal, so it's great as a hand sanitizer. All you have to do is rub it all over your hands, then give it a few seconds for it to dry, and you are in good shape in terms of getting rid of planktonic bacteria. You're not going to get rid of your biome with hand sanitizer, but you will get rid of any free living pathogens that may have gotten on your hands. Again, if you use it correctly. But there's always a but, right? If you develop a habit of applying hand sanitizer 20 times a day, that alcohol is going to dry out your hands. Remember, we said that alcohol can damage material over time. It can damage rubber. It can damage lenses. It can also damage your skin. So you have to learn how to use hand sanitizer correctly, and you have to use it when you're most likely to have been exposed to something, and try to prevent yourself from getting sick by touching your hands to your face. But you can overuse hand sanitizer, just like you can overuse soap. If you use hand sanitizer to the point where your skin starts to crack and even bleed, now you have opened up a pathway for pathogens to get into your skin. So the long answer can be summarized this way. Antibacterial soap is not going to harm you because all soap is antibacterial. You have to use it responsibly, though. You have to use it appropriately, because if you overuse it, you can damage your skin. And if you damage your skin, you have opened up a way for bacteria to gain entry. Hand sanitizer can also be overused. And if you use it to the point where your hands crack and bleed, you have opened up your physical barrier, and you can get bacteria into your tissue. I don't know how your skin reacts in the winter, but my skin does not react well to the winter weather. And because I work in laboratories all day, I wash my hands a lot. I wash my hands before I begin my work, and I wash my hands when I'm finished with my work, class after class after class. So my hands take a beating in the winter. And I have to be very careful. I have to be careful because I don't want cracking and bleeding on my hands, because I don't want germs, I don't want bacteria and other microbes to get in. You have to be careful. And sometimes that means washing your hands really well and then putting a little moisturizer back on your hands. In the winter, I try to keep a bottle of soap on the sink in my home and a bottle of moisturizer. So I wash, dry, and then put the moisturizer back. Yeah. Your skin is so amazing. Your skin is a physical barrier. It also contains lots of lovely natural chemicals that are antibacterial. Your skin's amazing. And your microbiome is amazing. Your microbiome protects you from a lot of illnesses. The problem is we have holes all over us, and viruses and things can get in and make us very sick. So Elaine is saying, yeah, people who work in healthcare, right? Especially in the winter. You see it all the time, right? Think about the physician who goes in and out of an examination room 30 times a day. That physician is, pumping the foam hand sanitizer on the wall when he goes in or when she leaves. And that's a lot. That's a lot of alcohol to be putting on your hands every day. You have to be careful because you don't want to damage your skin on any part of your body. You don't want to damage your skin if you can help it. Yeah, gel in, gel out, right? It's horrible on your skin. And yet it's very important. It's very important because these hands that we love spread a lot of disease. Remember, for like the common cold, it's not respiratory droplets that pass the common cold, is it? It's not respiratory droplets so much with influenza, it's your hands. Somebody who has the flu or who has a bad respiratory infection wipes their nose and then touches the doorknob. And then you come along and you touch the doorknob and then you rub your eyes. The hands pass a lot of infection, a lot of gastrointestinal infection, respiratory infection. Yeah. Elaine's saying there are some great barrier type lotions too. Yes, there are, Elaine. Now, some people don't like the barrier lotions because then when they go to wash their hands next time, like with the next patient, they feel like they're not really getting their hands clean. Again, maybe a barrier lotion once or twice a day. Again, you got to work it out for your own skin. The key is you don't want to let your hands get so dried out in the winter that they're cracking and bleeding. And I know it happens to me if I'm not careful. Kids are nasty. Yes, Valerie, kids are disgusting. Oh my gosh, there's always some material dripping out of them somewhere, right? I mean, and it's usually green, you know. Think about those poor daycare workers and those poor teachers who are constantly wiping noses all day long. But now that we've been talking about the immune system, remember, it's all kind of important, right? It's important. It's important that children's immune systems get exposed to things because that's how you build up immune memory. And that's how you build up the strength of your immune system. If you lived in a bubble and you never got exposed to anything, you never got exposed to soil, and you never got exposed to foods with their own natural bacterial communities, and you never got exposed to animals or other people, if you lived in that kind of isolation, you would be in a lot of trouble if you ever met a person because your immune system would be naive. It would not have ever met anybody. And while there are some immune abilities that you have right from the get-go, remember your innate immune system is just there and working all the time. It's important to build up your adaptive immunity over time. It's important. And that means live your life. Live your life. I don't encourage parents to, send their kids out in the backyard to spoon dirt into their mouths, but it's a perfectly natural thing for little children to put their hands in their mouth and up their nose. That's part of nature. That's part of not only them developing, but also part of building their biome. When a little baby, when an infant breastfeeds from its mother, it is taking in bacteria from the mother's skin. And that's an important part of why breastfeeding is so good for a baby. It helps build their biome. Yes. Yeah, kids can be pretty gross. Let's be honest. Kids can be pretty gross. I say that as a grandmother to two. Yes. My little granddaughter, who I shared with you, was premature. My little granddaughter came for a visit and did a perfect projectile spit up onto Nana and down the front of Nana's shirt. So, I mean, it was Olympic in its caliber, really. It was just a perfect trajectory of spit up this weekend. So, And then she just looked at me like, aren't you proud of me, Nana? Look what I did. Oh my goodness. So yes, all of it is very natural. And we need to remember that. We need to remember even during a pandemic, we are built, we are built to interact with the world. Our immune system is perfectly structured to interact with the natural world, and we need to allow it to interact. All right, very good. And I will say this, as you're going through the vaccination materials, as you're going through the introduction to vaccination lecture this week, I want you to really give some thought to what's happening in our country and around the world right now, because we have reached a point in the pandemic that is very strange. We have not hit herd immunity in our country, and we are unlikely to hit herd immunity, unfortunately. And that's because we live in a free society. Sometimes freedom is a double-edged sword, right? There are parts of the world where people will be vaccinated whether they want to or not. But in our country, because we have freedom of choice, there will be people who choose not to be vaccinated. So right now, there are swaths of the country that are at herd immunity and others that aren't. And right now we are seeing another wave of COVID. Likely because of July 4th and the fact that everybody probably got together on July 4th. So we're seeing massive uptake in case numbers and in deaths, but only in certain regions, which is so strange to watch. It's fascinating as a scientist, and it is horrifying as a human being to watch people get sick and die when we have a vaccine available. We have multiple vaccines. So keep that in your mind. Keep in mind while you're watching the vaccination material, especially the herd immunity part of it, because it is just, it's within our grasp right now to end this, but we can't do it. We can't seem to do it. It's tragic. I'm going to call it tragic that people are still dying of COVID because of unvaccinated, susceptible hosts. John, yeah, and John, that's such a good point. That's another really interesting thing is this idea of breakthrough infection. Remember that, and I apologize, I can't remember if I brought this point up but when these vaccines were being tested, and all of these vaccines have gone through clinical testing, that's an important thing to remember. If anyone says to you, for example, I don't want to get the vaccine because it's experimental, these vaccines are not experimental. These vaccines have gone through clinical trials. They are not 100% approved because they have not gone through all of the parts of the approval process yet, but they have had their clinical trials done. They are not experimental. But you've probably heard that, oh, people who have been fully vaccinated are testing positive for COVID. Those are called breakthrough infections. Those happen with all vaccines. But the important thing is this. The important thing is this. Those vaccines were tested for protection against severe disease, hospitalization, and death. And different vaccines were tested for different combinations of those endpoints. They are intended to prevent severe infection, hospitalization, and death. So there will be people who test positive for COVID. There will be people who get COVID, who get sick, who are fully vaccinated, but it is highly unlikely that they will get serious disease. There will be people, particularly people who have immune system problems. Think about the elderly, especially. who get pretty sick. They've been fully vaccinated. They get COVID anyway, and they get sick with COVID. Those people likely did not respond to the vaccine. Even the best vaccines that we have, the mRNA vaccines, have a 90+% effectiveness. So in 90 out of 100 people, they would have mounted a sufficient immune response to prevent serious disease, hospitalization, and death. That means 5 to 10% of people who get vaccinated will not mount an immune response to the vaccine. There's something wrong. There's something going on with their immune system. Maybe they're not old enough yet, although we're not really vaccinating little kids. Maybe they're sick with other illnesses and their immune system is too taxed. Maybe they're elderly and their immune system has started breaking down. So number one, there will be some people who will get COVID who are vaccinated because they didn't respond to the vaccine and they didn't mount an antibody response. But there will be other people who responded very well to the vaccine and who have lots of antibodies and who still get COVID. But those people will get a mild case of COVID, clinically speaking. Those people will be home, perhaps not feeling great, feeling like they have a cold or the flu, but they won't get hospitalized. They won't get on a ventilator. They won't die the way the unvaccinated do. So the latest numbers out of the CDC, and these came out just this weekend, 99.5% of hospitalized COVID patients and patients who are still dying of COVID are unvaccinated. So 0.5% of the folks who are seriously ill and dying with COVID are fully vaccinated. 0.5. It does happen. It does happen because vaccines fail, and also vaccines have specific endpoints. And the endpoints for these vaccines were not complete prevention of infection. They were prevention of severe disease, hospitalization, and death. It's so sad. It's from just an emotional standpoint and a societal standpoint, it is very, very sad that people are still dying every day of COVID-19 in our very wealthy, very privileged country. But we are a free society. And unfortunately, this entire pandemic became very politicized, and the amount of misinformation out there is horrifying. It's horrifying. I hear it every day. I hear it every day. Folks who believe the strangest things about the vaccine. So, yes, John, it's a lot like kennel cough. Yes, the kennel cough vaccine is a decent vaccine, but it's a vaccine that protects dogs against this most serious disease associated with Bordetella infection. So it is possible for a dog who gets vaccinated for kennel cough to get a mild kennel cough. Plus, the kennel cough vaccine doesn't last very long. It needs boostering quite frequently. We usually booster dogs before they go into the kennel because it just doesn't have the memory associated with it that other vaccines do. Yeah, so only time is going to tell where this pandemic is going to go for us and for the world. But sadly, it does not look like the United States is going to reach herd immunity by the fall and the winter. And that, of course, is the time that infectious respiratory disease spreads most easily. Once we all go inside for the winter, that's when infectious diseases, especially respiratory diseases, tend to spread. If we don't hit herd immunity by October across all 50 states, we're in trouble. We're in trouble. This virus doesn't care. This virus doesn't care where you live. It doesn't care who you are. doesn't care what your political affiliations are. This virus is looking for a susceptible host. And if you are not vaccinated, you are a susceptible host. So it breaks my heart. It really does. Elaine, that's a very interesting question. And I'll answer this one and then we'll switch over to our assignment. Elaine saying, I worry about it being a bad year for lots of infections, right? Not just COVID, but also the flu and bad colds and respiratory syncytial virus and strep and all of it, right? Interesting fact, Elaine, is that last winter, the winter of, COVID and the winter of the pandemic, we didn't see a lot of flu, did we? And we didn't see a lot of common colds and we didn't see a lot of RSV. And there were a couple things that work there, a couple of reasons why the flu numbers were so low last year. First and foremost, we were separated. Humans were not congregating inside during the winter months. And so influenza had a hard time spreading from person to person because we weren't together. So those things don't spread very well unless people congregate. That was the first reason. But the second reason is also fascinating. The amount of SARS-CoV-2 virus was so high, just the physical number of particles of SARS-CoV-2 was so high that it literally just outcompeted influenza. It outcompeted all the other viruses. There was no room for them to get in because there was so much SARS around. These are particles of material, these viruses. They are physical particles of material. And as long as there are people who are susceptible to them, they will be replicated hundreds and thousands of times over. So every time a person got COVID, they spewed out thousands of more particles of SARS, and it literally just overwhelmed the other viruses in terms of access to hosts. It was kind of fascinating. It was a fascinating year in terms of what was happening with other infectious diseases. Yeah, I worry too. I worry that when we get back together this winter, it's going to be a bad winter. And now that we've really separated into two populations, the vaccinated and the unvaccinated, the SARS-CoV-2 particles will circulate among the unvaccinated. And the rest of us will be our usual susceptible selves to the other viruses. And like you say, we may end up with just a mishmash of everything this winter. And that's going to make it very hard from a clinical standpoint, because if a patient comes into the office with a runny nose and coughing and feverish, you got to think of COVID first, but it could be flu, or it could be a cold, or it could be another virus, and it's going to be very difficult. It's going to be a tough winter. So be prepared for everything. That's what I always tell students, healthcare students, be prepared for everything. Be prepared to have to mask up. Be prepared to maybe go back home for a while. Be prepared to be back, at school. Be prepared to be remote. Be prepared for everything, because who knows what's going to happen. So Elaine, with regards to children right now, the clinical trials are almost over for the 5 to 12-year-olds. So it's likely that by maybe October, if we're hopeful, by October, it's likely that the 5 to 12-year-olds will be able to get vaccinated if they choose. The clinical trials for birth to 5 are not even close to being over yet, so they probably won't be eligible for vaccination this winter. But the 5-year-olds and above ought to be, but it'll be close. It'll be like October, November. So what's very interesting about the Delta variant the newest variant. And remember, as long as this virus is allowed to circulate among susceptible hosts, we're going to get more and more mutations. So one of the interesting things about the Delta variant is that younger people are showing more susceptibility to it, and the symptoms are different. The symptoms are different in the little ones and in the adults who are coming down with it. A lot more upper respiratory symptoms now. A lot of runny nose now. That was not a primary COVID sign last winter. So that's the other thing. You have to be aware of changing symptomology with changing variants. Different age groups might respond differently to the virus. Yeah. You just want it all to end, right? You just want it all to end. But the only way for us to end it is to reach herd immunity. And we're not there. We're just not there. All right. And on that cheery note, I'm going to go ahead and pull up our assignment. So if you click on the instruction sheet, this is what you're going to see. There's some background information here, which kind of explains this whole concept that we use this whole range of different tests in order to help us identify microbes in the laboratory. And then we have to go to source material to find out which genus and which species have these particular test results that we would expect. There is one source that is considered sort of the gold standard of all of this, and it's a book called Bergey's Manual. Bergey's Manual, there's actually several different Bergey manuals, but Bergey's Manual of Determinative Biology is considered sort of the gold standard of source materials when you're looking for the laboratory traits of different bacteria. There are all kinds of charts and graphs and flow charts in Bergey's that help us to use test results to identify bacteria. Now, one of the things that you'll find in Bergey's Manual are these things called dichotomous keys. Dichotomous, if you translate that word, it just means two paths or two pathways, two choices. And when you look at a dichotomous key, it essentially looks like a yes-no kind of flow chart. It asks a series of questions, and you're going to have one of two choices as an answer. And so you follow the pathway down the chart, depending on which answer that you get for your particular unknown microbe. And at the end of a dichotomous key, you're going to get an identification. You're going to get a genus and a species of bacteria named. But, and this is very important, at that point, you have a preliminary identification, I'm going to show you the dichotomous keys that we'll be using for this assignment. But you're going to be using those keys to find out your preliminary identification of your unknown microbe. And then you'll have to go on into the internet to different sources to get a conclusive identification. So each student gets their list of laboratory test results. And again, half the class is going to get one set of test results and half the class is going to get a different set. And you're going to have to go down that set of test results and interpret what they mean as part of the assignment, using all of your lab notes that you have gathered this semester, and also using your textbook and any other source materials you need. You're also going to be provided with a dichotomous key. There are two of them posted for us to use. One is for gram-positive bacteria, and one is for gram-negative bacteria. So one of your test results is going to be the results of gram staining. And from that result, you'll know which key that you need to go to You'll then use some of your data as you go down the flow chart, as you go through the key. Some of the test results will help you work your way through the dichotomous key, and that will provide you with a preliminary identification. But your data set is big. It's got, I think, 16 or 17 test results in it. You're only going to use maybe 4 or 5 of those on the key. So you have lots more data that you will want to use in order to make your final definitive identification. And that's where you're going to want to go to the internet and do a little additional research, because there are lots of source materials out on the internet that can help us doing this kind of work. Now, I do ask that you go to what I've written down as reputable websites. So websites that have a good reputation, that you wouldn't know as a student which ones have a good reputation and which ones don't. So I've listed the top 3 for you that I think you'll find very helpful. One's called Microbe Online, one's called Microbiology Info, and one's called Microbe Wiki. Those are 3 sites that have good reputations for accurate information. You may also find other websites that you decide to use. And if you have any questions about whether or not you have found a good website, a reputable website, you're free to contact me, message me, and I'll let you know if it's a good one or not. The one website that I don't want you to use for the assignment is Wikipedia. Okay, so don't use Wikipedia for this assignment. Why would I not want you to use Wikipedia? Why would I not want you to use Wikipedia? Does anybody know? What is Wikipedia? How is Wikipedia produced? What does it mean when a website uses that word wiki? Yeah, Valerie's got it. It's open source. Anybody can add to Wikipedia. And the idea is that people who have knowledge will come in and they'll put information in. But unfortunately, it also means that trolley kind of people can come in and put misinformation in. Plus, there are some people that think they're putting accurate information in who are not putting accurate information in. So Wikipedia can be a great resource, but it can also be a disastrous resource because you don't know how good the information is. Now, Microbe Wiki is also a wiki site. It can also be added to by individuals, but it has some restrictions as to who can add to it and who can't. So it's generally has a much better reputation for the information that's on it. Okay, so that's the one source I don't want you to use is Wikipedia. I would definitely use those 3 that I've given you first. And if you still need to find a particular test result or you still have a particular question, you can go to other websites. Okay. So here's the actual assignment. Using the test results that are being provided to you and those dichotomous keys and your textbook and any other reputable internet sources, you are to identify the genus and the species of the unknown bacterium that you were assigned, and then answer these questions. You're going to type your answers directly on this handout. All right, this is important. I want you to type your answers directly on this handout. Don't make an independent answer sheet for yourself. You can use as much room as you need for each question. You're going to be graded on 2 things. You're going to be graded first and foremost on the accuracy of your answers and the thoroughness of your answers. How complete were you? How thorough were you in your explanations? About 80% of your grade comes from accuracy and thoroughness. The other 20% is going to come from the quality of your writing. And what I mean by that is you are expected to use complete sentences and paragraph form and correct spelling and correct grammar. So make sure that you check and double check your answers before you submit them. if you need to have someone else proofread for you to double check your sentence structure, of course, do a spell check, do a grammar check, okay? 20% is the quality of your writing. You are at a point now in your education where everything that you write should be written correctly, should be written professionally. And this is a good opportunity to practice that. So question 1 is 35 points. This is the bulk of the points for the assignment. All right. For each of the test results that you were given, indicate how you interpreted the results. Be as complete and specific as possible. All right. Start by stating what the test is used for, and then interpret the specific result, describing what the test result indicates about the characteristics of that microbe. Okay. I've got some examples for you of what I mean by this. What I mean by first state what the test is used for, and then interpret your results. All right. Here's an important thing right here. Do not attempt to identify your bacterium while you are interpreting the test result. Don't do that. All you're doing is interpreting it generally. You're going to identify your bacterium in the next question, not in question 1. So here's examples for you. Let's say the first test result that you get in your data set is that on a wet mount, the cells were motile. All right. First, tell me what the test is used for. Wet mounts are used to examine living microbes, including whether or not those microbes are motile. Okay, that's my way of saying what a wet mount is used for. You're going to say something similar, but you're going to say it in your own words. Now, interpret this test result. The test result is that cells are motile. What does that mean to you? What do you now know about your unknown bacterium? This result indicates that the unknown bacterium is motile, and therefore it has at least one flagellum. That's an important piece of information to know, right? Now you know something very important about your unknown. It has flagella, because that's the only way it could be motile. Remember, bacteria don't have cilia. Bacteria don't have cilia. Eukaryotic cells have cilia. Bacteria have fimbria, pili, flagella, and flagella is the one that helps them move. Okay. Let's look at another one. Gram staining showed a pink-colored rod-shaped cell. That's your result. Tell me what that test is used for first. Gram staining is used to determine the gram status of bacteria. The gram status is a description of the thickness of the layer of peptidoglycan in the cell wall, as well as the shape of the cell. That's what the test is used for. Now interpret your result. Now interpret the pink color and the rod shape. This result indicates that the unknown bacterium has a gram status of gram-negative rod, or I could have said gram-negative bacillus. Like all gram-negative bacteria, this unknown has a thin layer of peptidoglycan in its cell wall, surrounded by a second outer membrane. You now have a second really important piece of information. Your unknown is gram-negative, and it's rod-shaped, and it has a flagellum. So you've already ruled out all the gram-positive bacteria. You've already ruled out all the gram-negative ones that are spirochetes. You've already ruled out all the gram-negative ones that are cocci. You've got two really important pieces of data there. The unknown has a gram-negative status, it's rod-shaped, and it has a flagellum. Okay, you're building information to help you identify. Let's look at another example. Remember, some of our testing involves special media. And there's going to be a couple of test results in your data set that use special media. And these are special media that you have a little experience with because we used them in our laboratory. So test result 3, grew on EMB as purple colonies, no green metallic sheen. First, tell me why we use the test, then interpret that test result. EMB, or eosin methylene blue, is used as a selective and differential medium. It selects for gram-negative enteric organisms. It differentiates between bacteria that can ferment lactose and or sucrose, and those are called coliforms, from those that can't ferment lactose. It is also used to differentiate E. coli from other fecal coliforms, right? We can know if we have E. coli based on EMB. Now interpret this. Interpret that it grew on EMB, it grew as purple colonies, and there was no green metallic color. The purple colonies of the unknown bacterium indicate that the unknown organism is a coliform, and the lack of green metallic sheen indicates that the coliform is not E. coli. It's something other than E. coli. You got another really big piece of information there. Okay, you've got a coliform. You've got a bacterium that lives in the intestine, and it can ferment lactose, but it's not E. coli. That's very helpful, right? It's not E. coli. Remember, in addition to special media, we also looked at oxygen category, right? We looked at thioglycolate broth, and that was a test that helped us put our microbe into an oxygen category. So your result in this example is in thioglycolate broth, grows throughout the tube, but most growth occurs in the oxic zone. Okay, first tell me what the test is used for. Then tell me how you would interpret this. Thioglycolate broth is a biochemical broth used to determine the oxygen category of an organism. Now, do you have to say that exact thing? No. Maybe you're going to say thioglycolate broth is a broth that uses agar to make it semi-solid. And sometimes it has an indicator dye in it. I mean, you could choose any way to explain that. Tell me what the test is used for. You're going to put it into your own words. What the test is used for, why we bother using it, and how you interpret the results. You get to pick and choose what information you think belongs in that answer. Knowing that you got to be accurate and you got to be complete, you got to be thorough. Okay? Now, how would I interpret this? Oh, I wrote a little bit more here. I wrote it's semi-solid, it creates an oxic zone and an anoxic zone and a gradient of oxygen in between. This result indicates that the unknown bacterium is a facultative anaerobe. Facultative anaerobes do not require oxygen to make ATP, but they prefer to use it if it's available. They grow best in the oxic zone, but they're also able to grow throughout all other regions of the thioglycolate tube. You only need to write, about a paragraph for each one of these. You don't need to write a dissertation about thioglycolate broth. You can tell me why thioglycolate is used, and you can interpret your test results in a good paragraph. Okay? So don't write a single sentence. It's not possible to write a single sentence, is it? Because you got to tell me two things. You got to tell me what it's used for in the lab and then interpret your particular result. Write me a good paragraph. Now, you're going to do this, something like this, for each one of your test results. And you've got, again, I think 16 or 17 test results. So you're going to write your answers right on the sheet. So under question 1, you're going to type in your answers for each individual test, what the test is used for, what your particular result means. Now, question 2 is worth 10 points. Question 2 says, according to your research, what is the genus and species of the unknown bacterium that you were assigned? Be sure to use the complete name and be sure to use the correct formatting now that you know how to do that. And write it in a complete sentence, please. So here you are going to write one sentence. My unknown or the unknown bacterium is, and with the genus and the species. The full complete name written correctly. Let's review what that means. We can type out a scientific name by either italicizing it, that's the way most people do it, or you can write it in regular type and then underline each of the names. The genus name gets a capital letter. The species name gets a lowercase letter. Make sure you spell it correctly. Make sure you've written it in the right format. Full name, complete sentence. Now, this question is worth 10 points. And one of the most common questions I get from students is, what if I get it wrong? What if I get it wrong? What if I end up with the wrong identification? If you've made your identification based on the data that you were given, and you made maybe one mistake, I'm not going to take all 10 points off. But if you made an identification that doesn't make any sense based on your data, I'm going to take all 10 points off, okay? But here's the thing. That actual identification is only 10 out of the 50 points. What's most important is that 35-point first question. Make sure you interpret, make sure you write out those interpretations accurately, thoroughly. That's where the meat of this assignment is. Show me that you understand what we talked about this summer and that you've learned about these tests. And if you get the actual genus and species wrong, I will base how many points to take off based on how you used your data. Does it make sense how you used your data? Okay. Remember, as you're answering each test in that first question, don't say to me, this means I have. Don't do that. Don't identify your microbe in question number 1. You can't. You need all of the data to identify your microbe, okay? So question 1 should be just left generally the unknown. This means this for the unknown. Don't tell me, oh, this means I must have this, all right? Save that for number 2. Now for number 3, for 5 points, you're going to cite all of the source materials you used for me. You don't need to cite, this is important, jot this down. You do not need to cite your class notes. You do not need to cite the dichotomous keys that you were provided with. If you use your textbook, please tell me, please cite your textbook. If you use internet sources, which you will need to, please cite those. Now, your citations don't have to be presented in any specific style. In other words, you don't have to use AMA. You don't have to use any particular formatting style. for your citations. But I need to be able to locate the specific material you used by using your citation. So if you used this website, let's say you used microbeonline.com, make sure that the URL that you cite takes me to the exact page that you used, the specific material that you were looking at and using in your answer. Okay. Also, please do not simply copy and paste URLs. Put them in the same font, the same size as the rest of your work. Remember, professionally, any work that you do, you want it to look as good as the quality of the work. So when you hand something into a supervisor, for example, a report, let's say, you don't want your citations to be in 15 different fonts or 15 different colors or 7 different sizes. Select all of it, put it all in the same font and the same size as the rest of your work. All right. How your report looks reflects upon you. Your supervisor is going to be more impressed with your work if it's well written and it has a professional look. All right. So keep that in mind. So 3 separate questions. Question 1 is worth the most. A separate paragraph for each test result that you're given. Question 2, a single sentence, the identity of your microbe. Question 3, your source citations. Okay. You're going to type directly on this handout, so you're going to need to download it from Canvas. Now, let's take a look at these dichotomous keys, because remember, the very first thing after you work through all of your tests and you interpret all of your tests, now you need to go to the dichotomous keys, and you're going to use your gram stain result, because there are 2 keys. There's one for gram positive and there's one for gram negative. So according to that example I just gave you, my unknown is a gram negative. So I would use the gram negative dichotomous key. All right. Let me pull this up for us, if I'm capable of it, which I may not be. Hold on. Come on, keys, come on up. All right. When you click on where it says dichotomous keys on Canvas, this is what you'll get. It's a Word document and it has 2 images in it. These actually come out of the lab manual that we typically use for this course. So it's a little hard to see, so I'm going to zoom in a bit. But this first image is for gram-positive bacteria. Can you see that? And the second image, if you scroll down, is for gram-negative bacteria, right there. So this is what a dichotomous key looks like. It's a flow chart of choices. And you're going to follow along the diagram based on which choices you make, which test results your unknown had. And again, these keys are not going to use all of your test results. There's only a handful of them that show up on this chart. That's why you also need to go to internet sources. If you know that your unknown microbe is a facultative anaerobe, you're going to need to go to the internet to double check your preliminary identification. This chart is going to give you a preliminary identification. Let's say that your preliminary identification was Staph aureus. That's a gram-positive organism. And you know from your data set that, and I'm making this up, that your unknown grew on MSA and turned the media yellow. You're going to need to find a source somewhere, right, that says, oh yeah, Staph aureus does that. Staph aureus will grow on MSA and turn the media yellow. Okay. Let's say you find that the phenylalanine deamination test in your data set, it says that the phenylalanine deamination test caused no color change. And that means that your unknown microbe was unable to deaminate phenylalanine. It wasn't able to break that amino acid down because it didn't have the enzymes it needed to do that. You're going to need to find a source somewhere that says, yes, Staph aureus should not be able to deaminate phenylalanine. That's a confirmatory test result. So you're going to need to make sure that you find out, based on your preliminary identification, make sure that you find a source that says, yes, your preliminary identification looks right because it should have a negative on this test. It should have a positive on this test. It should turn green on this test. It should turn blue on this test. You should see yellow on this test. There shouldn't be any gas in the Durham tube. You want to go down the list, and you're going to have to go to the internet to find source material that confirms your preliminary identification. So let's say your test results indicate that your unknown organism is gram-negative. You can see from this particular dichotomous key, the next thing you're asked is the shape. If you have a coccus, you're going to go this way on the flow chart. If you have a rod, you're going to go this way. So we'll go this way since my example was a gram-negative rod. Now what do I do? Well, now it says oxidase. Now it says oxidase. But look, when you travel these around, when you travel these lines, here's positive. Can you see that? Here's negative. So you have to use your oxidase test results. And let's say, and I'm making this up, let's say for my unknown, mine was oxidase positive. So I'm going to go this way on the chart because there's the positive. See it? If mine was oxidase negative, I would go this way. I'm going this way. The next one says glucose oxidized. What does that mean? What test do I go to for that? Glucose oxidized. What test could I go to for that? Glucose oxidized. What test that we have been talking about recently had to do with whether or not a microbe can do oxidative catabolism of glucose? Elaine's got it. Yeah, the OF test, the oxidative fermentative test, told us whether or not a microbe could do oxidative catabolism or fermentative catabolism. Yeah. So let's say in my data set, it said positive. Let's say that my open air OF tube turned yellow. So I know that my microbe is able to do aerobic oxidative catabolism. So I'm going to follow this way because that's where the positive is. Now, if you look, it says growth at 40 degrees C. That seems a little odd. We didn't talk a whole lot about incubator temperatures. We said most microbes most microbes grow between 35 and 37 in the incubator. Okay. There will be a little note in your data set about whether or not your microbe has any unusual growth temperature characteristics, okay? Because these particular dichotomous keys use that information to help you differentiate. I want you to look at that particular data. If it's capable at growing at 40C, which again is a little warm, look what it leads you to as your preliminary diagnosis, Pseudomonas. In fact, either one would be Pseudomonas. It's just different species. Pseudomonas aeruginosa is the one that we used in lab this semester. This was one of our experimental organisms this semester. Pseudomonas fluorescens we did not talk about, but Pseudomonas aeruginosa can tolerate a little more heat in the incubator than its cousin Pseudomonas fluorescens can. So that's why that result is there. Now, I want to point something out to you on both of these keys. You'll notice that a lot of the names of the organisms have been circled. Ignore that, please. This is my particular key that came out of my particular copy of the lab manual. And so I was doing a different exercise during the course of the semester, and I was circling certain microbes. Just ignore the circled names. They don't mean anything to you, okay? You're just going to use this flow chart as best you can to get to an organism at the bottom, and that is your preliminary identification. You're going to use the test results from your data set, again, 4 or 5 of them for each of these charts, to get to your preliminary identification. And then from your preliminary identification, you're going to go to the internet. You're going to say, well, I think I have Bacillus megaterium. I'm going to use all the rest of these test results to confirm that. I'm going to make sure that Bacillus megaterium is oxidase positive and catalase negative, and the OF test turned yellow, and it grew on EMB, and you know, you're going to make sure that all of your test results make sense for the organism you think you have. And if they do, great, you've made your confirmed identification. If they don't, then something went awry. You're going to want to go back to the key and look again. Okay. So first thing you do, interpret all your test results, interpret your data. Know whether you have a positive or a negative or what the test means. Then find the right key to use based on gram status, and then make your preliminary identification. And once you think you know what you have, then use all the other test results at your disposal to confirm it by going to the internet and to your textbook if you find that helpful. Okay. And you'll get a chance to do what happens in labs every single day. Microbiology labs all the time. Okay, so a couple of you are indicating in the chat that you are interested in working with classmates, yeah? So there's some information here that people are leaving. It's like email and phone numbers. This is what I'm going to do. I'm going to set up a discussion board that is dedicated to this, the unknown microbe assignment. So you can check in on that discussion board. You can put out your email if you're happy to do that. You can put out your whatever, your contact information, or you can simply just type to each other on the discussion board. All right. Hey, my last name starts with M. You know, I'm interested in connecting with other people who have that data set. You can talk to each other over the discussion board, in other words. What did you find out for the OF test? What source did you use? for the, I don't know, the nitrate reduction test? Did anybody find a source for this test? Okay, so you can talk to each other, you can communicate with each other over that discussion board. I'll set that up right after class today. Okay. So you can get started on that as soon as you have time, and you'll want to get that submitted to me by Sunday at midnight. Now, that is a hard deadline. What do I mean by that? I can't accept your work late on this one because Monday is the final exam. Okay, so be sure to get that unknown microbe assignment to me either before or up to Sunday at midnight. As soon as you have it ready, send it to me. And if any questions come up between now and the final exam, any questions about anything, about lecture material, about the lab practical, about the final exam, anything, message me, okay? Message me over Canvas, because that gets it up high in my inbox, and we'll figure it out, okay? Any questions? And let me say, while I do have you, since I won't get to see you in person again, let me say that I've really enjoyed working with you this summer. You've been a great group. I'm sure you can imagine that I get a huge mix of students in a course like this. And some students work harder than others. And some students don't work as hard. And I've been so pleased and privileged to work with you guys this summer because you've been a really great group. And I'm wishing the best for all of you in your future endeavors. I'm sure you'll be successful because you really are just a top-notch group of students. So, all right, everybody, stay in touch if you need me. I will stay in touch with you over the announcements feature. All right, everybody, take care.
Thank you.